Okay, so second up, we have Dr. Alana Mckay. She's going to be talking about iron status and regulation of absorption in elite athletes. So Dr. Mckay completed her Ph.D. at the University of Western Australia in 2020 and has since been working as a postdoctoral research fellow at Australian Catholic University.
Okay, thank you. So hi, I'm Alana. It's really nice to be presenting in front of real people. It's been a really long time for me coming from Australia. So today I'm going to follow on from what Jamie's really spoken about, some of the supernova studies, and take a different angle being the iron stuff, which is what I'm really interested in. So before I started, I didn't think anybody would actually know who I am. So I just put a quick background. I am from West Australia. That was a really nice intro. I did a Bachelor of Sports Science. And then following my bachelor's degree, I went and did a one-year internship style year at the Australian Institute of Sport. And then I ended up going back to study after thinking I wasn't going to do that, and did a PhD looking at iron metabolism and dietary manipulation, which Jamie, who I was annoyed, really bad time to get COVID, he actually marked my thesis, which was really great. And then after my PhD, which was embedded both within the AIS and also the West Australian Institute of Sport, I landed at ACU with these guys as a postdoctoral research fellow. And now, even though I'm a real, technically a full-time academic, I'm still very much embedded as a sports scientist, and all my projects are very applied. These guys are definitely the molecular guys in the group, and I'm a bit more on the applied sports science side of things. So I wanted to talk today about iron. It's definitely my sort of passion. And I thought, firstly, why is iron important? So firstly, it's really important for erythropoiesis, so the production of new red blood cells. If you think of a red blood cell and hemoglobin, which is a major component of red blood cells, heme literally means iron. So in order to improve our VO2 max or aerobic capacity, we need to increase our red blood cell number, and therefore iron's a fundamental building block of that. We often also correlate iron deficiency with neurological deficiencies. This is less studied within athletic populations, but it could be suggested that iron deficiency may lead to decision-making impairments in sport or differences in reaction time. Energy metabolism is also dependent on iron. So within the pathway to produce ATP, iron's an essential molecule, and therefore athletes that are iron deficient could be considered metabolically inefficient in some instances. And we also know iron's really important for immune functioning, which is really what my PhD looked at. Both iron deficiency and iron overload have separate effects on the immune system, both equally detrimental. And if an athlete's constantly sick, we see they're not able to train and compete to the best of their ability. So despite iron being really important in athletes, about 3 to 10 or 11% of males are often seen to be iron deficient, and anywhere between 15 and 35% of female athletes. And that greater incidence of iron deficiency in females is attributed to the iron losses or greater iron losses that occur as part of the menstrual cycle. So when we speak about athletes more specifically and their risk of iron deficiency, it comes down to 3 key factors. So the first one is that they have an increased iron requirement. If you're going to increase your VO2 max, you need more red blood cells, and therefore you need more iron to do that, for that process to occur. We often also see increased avenues of iron loss that occur with athletes. So sweat, you can imagine an athlete's probably going to sweat about 10 times more than somebody who's sedentary. Hemolysis, which is sort of heel strike with running. So every time the heel may hit the ground, you can see a destruction of the red blood cell, which releases iron into circulation, which is no longer used for storage unless it's reabsorbed. And also gastrointestinal bleeding. So particularly for endurance sports, it's quite common, and that forms a direct loss of iron. So cumulatively, these can provide it's not the most significant way of losing iron, but can still contribute to an athlete's having higher risk of iron deficiency. But the major component or reason we think athletes are more iron deficient than the general population is because of the action of the iron regulatory hormone hepcidin, which reduces an athlete's ability to absorb iron. So really briefly speaking, when we exercise, we start to deplete our muscle glycogen stores, and that acts as a signal for the muscle to release IL-6. And that acts as sort of a mitokine to be able to get the liver to produce glucose from either glycogen or any other substrate. And that glucose can be fed back to the muscle to continue exercising at the same intensity. But what we also know is that the liver that IL-6 signal upregulates a pathway called STAT3, and that causes hepcidin to be produced. When hepcidin is produced, it actually binds to the iron transport sites at either the enterocytes, the macrophages, or the hepatocytes. And what it does is by binding to those sites, it blocks iron absorption. So when hepcidin is elevated, we see a decrease in iron absorption and plasma iron concentrations, which is a likely contributor for iron deficiency in athletes. So this study was done by Pete Peeling, who was my PhD supervisor a long time ago now, and he simply tracked the time course of changes in both IL-6 and hepcidin in response to exercise. So we had 8 moderately trained endurance runners, and they simply exercised for 60 minutes and also did a sedentary trial, but I've just presented the exercise trial here. And they saw that IL-6 increased immediately post-exercise, and then 3 to 6 hours later, there's this increase in the iron regulatory hormone. I talked about hepcidin, and therefore during that period or that post-exercise period, there's a likely time frame in which iron absorption is actually impaired. And if we think of it in an athletic context where they might be training multiple times a day or having really big exercise bouts, that period of impairment can go on theoretically for a long time. So more recently, we've had Nikita Fensham, who's a PhD student of ours at ACU, look at this in a more athlete-specific context or in the way that athletes train. So we had 16 elite male rowers from the National Olympic Training Centre, and we had them do a randomised crossover design. So separated by 7 days, they did one exercise trial where we actually gave them calcium supplementation and another exercise trial where we gave them a placebo. And so the point of this isn't really looking at the calcium or placebo results. I'll save those for another day. But during each exercise trial, the interesting part was we did 2 exercise sessions. So they did a 90-minute rowing ergometer session separated by 2.5 hours. So one at about 9 a.m. and one at about 11:30. And this was done at T1 power, which is a really niche rowing specific intensity, but is about pulling 260 watts, so 145 beats per minute heart rate, RP of about 12. So steady state exercise. And what we saw was, we saw the gray bars there represent the exercise bouts, and we saw IL-6 increased after both of those exercise bouts. So it's like a double peak in IL-6, and the 2 lines there are the 2 trials. But again, just look at the sort of time course of the changes. But what we saw with hepcidin is hepcidin increased in that 3-hour period as we expected it to, but it actually just plateaued 3 hours into the next exercise bout. So what we've sort of seen is that despite the double peak in IL-6 resulting from the double training day, hepcidin just peaked and remained elevated. And it might suggest that when we're looking at iron absorption, it's more the timing rather than the number of sessions an athlete's doing that is going to be the most important factor when looking at iron absorption. So if an athlete's doing 3 sessions a day, it might actually be comparable to an athlete that's just doing 1 session if it's done in the morning. And that we think links in with some of the diurnal variations that we see in the hormone more generally. So when we look at correcting an iron deficiency, we often have 3 treatment modalities, and they often depend on how severe the iron deficiency is. But today I'm going to talk about the dietary intervention aspect and link in some of the work that Jamie's just talked us through. And instead of talking about how I may correct an iron deficiency, I might point out some of the ways that I would not go about treating an athlete with an iron deficiency. And the first one is via a ketogenic, low carbohydrate, high fat diet. So when we look at dietary iron, the current recommended daily intake for iron is about 8 milligrams per day for males and 18 milligrams per day for females, with the higher female intake, again, attributed to their higher iron requirements. These are, before we go any further, very much specific to the general population, and we have no athlete guidelines at this point as to how much iron they should be consuming. Even though we know that their iron requirements and their iron losses are likely greater, we still don't have a figure which we should be targeting for athlete populations. So in one of the studies that Jamie talked about, which was the first supernova, we implemented a 3-week ketogenic diet. And what we did following the studies, we actually went back and looked at what was going on with iron. These diets were formulated with dietitians to meet energy and macronutrient goals with a more holistic approach to the micronutrients. So we went back and saw what was happening. And what we saw was that the low carb, high fat diet had a significantly lower amount of dietary iron on average each day. And when we looked at where those sources of iron were coming from, we actually saw that the more bioavailable forms of iron, being heme iron, which is animal-based products, So red meat, poultry and fish, there weren't any differences between the diets. And it may be because we actually are really quite strict around matching protein between these two diets. But the biggest sources we saw were that bread, cereals and grains, which are carbohydrate-rich foods, when they're eliminated from the diet, they eliminated a big source of dietary iron from the diet as well. And there's been another publication since our initial publication, which this time looked at a self-selected diet in athletes over a 12-week period, and they saw quite remarkably similar findings in that the dietary iron contents of the diet were much lower. So mean daily iron intake was significantly lower in the ketogenic group. However, it was still above the recommended daily intake of iron. But again, those intake values are very much specific to the general population and not to athletes. Now, we also think that the ketogenic diet can influence iron regulation. So I showed this slide before, but if we think about where carbohydrate restriction fits in terms of a ketogenic diet, we're going to see an accelerated or even greater reduction in muscle glycogen storage, which is going to cause an increased signal of IL-6 and potentially increased hepcidin and potentially decreased iron absorption. And if you think about that in a more chronic sense over especially timeframes where people are implementing ketogenic diets, it could be a pretty big contributor to iron deficiency. So this was data from Supernova 1 and 2. We had athletes come in. We had 50 athletes in total, and the first 3 days we had them all on a high carbohydrate diet. And then we did a bunch of baseline testing, and then we allocated them to either continue on the high carbohydrate diet or to start adhering to a ketogenic low carb, high fat diet. So this diet was less than 5% carbohydrate, so about 50 grams per day, as Jamie said, and predominantly made up of fat. And at the end of each of those two dietary blocks, we had the baseline and adapt testing phases, which included a 25-kilometer race walking protocol. So see whether an athlete's iron deficient. So at baseline, we saw that both groups are really similar, but after the 3-week training intervention, we saw big reductions in iron status, which firstly just shows that training can have a huge effect on your iron storage if you're trying to mobilize iron for adaptation. But we actually saw the biggest reduction in the carbohydrate group, which is sort of alternate to what we were maybe thinking, particularly considering the carbohydrate-rich diet had more iron in it, and also given the pathway we thought that may impair iron regulation from the ketogenic diet. So we had two hypotheses. First, that we thought that the greater decrease in iron in the high carbohydrate group, despite the higher dietary iron intake, may be indicative of the greater adaptation. So lower iron storage doesn't always mean iron loss. It might have meant that iron was being utilized for new red blood cells. And some of the data that Jamie presented around the performance outcomes and the race benefits seen to the high carbohydrate group may support that hypothesis. The second is that serum ferritin is an acute phase protein. So it may have just been artificially elevated under conditions of increased inflammation. But we have no data to support that hypothesis. That's simply us looking at some of the inflammatory pathways that have been reported in the past. What we did see is that IL-6 response was significantly greater. So the post-exercise response after adhering to the ketogenic diet. But the hepcidin data wasn't as convincing. We did see significant differences between the diets, but they didn't come out in the p-values just when you looked at the confidence interval. So it's not particularly convincing, but there is the possibility that those differences in ferritin were influencing hepcidin. So given hepcidin's responding to iron storage, there may have been a confounding factor in this study. Sorry. So we actually repeated this study as part of the Supernova 5 study that Jamie talked about. So this time we implemented just a 6-day dietary intervention. And what you can see is after 6 days, it was too short of a time frame for any differences in serum ferritin to change. So almost we've nullified that potential confounding factor. And what we saw this time is that it was really clear that both IL-6 and hepcidin were elevated as part of the response to a ketogenic diet. So as a take home, we can see that the low carb, high fat diet can be low in dietary iron, which may over time place an athlete at an increased risk of iron deficiency. We are seeing inflammatory responses to exercise are increased after adhering to the low carb, high fat diet. And this may actually have potential implications not only to iron, but other body systems. So we've been doing some work in the bone health space, which we know can be affected by IL-6. So it may be a bit more multifactorial than just what I've presented here. And we also now have evidence that hepcidin is increased after short-term adherence to a ketogenic diet. So there may be implications for reduced iron absorption over time, particularly from foods and supplements, which may place an athlete at a greater risk of iron deficiency if adhering to for long time frames. And as Jamie said, there's a lot of talk that this isn't something you do for 1 week to 4 weeks. It's something you would adhere to for 6 months. So the second dietary approach I wanted to talk about was low energy availability diets. And Jamie's done a fantastic introduction of that for me, so I don't need to cover off what that really is. But essentially it's a highly caloric restricted diet. So when we look at the literature currently for evidence that link low energy availability and iron deficiency, we've seen that the REDS consensus statement, which is relative energy deficiency in sport, lists 10 health consequences from adhering to low energy availability, one of which is hematological dysfunction, which covers off iron deficiency. There's two sort of arguments at the moment, whether it's REDS or whether it's the female-male athlete triad. But no matter what way you look at it, both syndromes have been marked with studies that are looking at low iron status, and not really sure why and how iron has linked into those up until this point, particularly when you look at the more scientific evidence. So a lot of the talk is a bit lay and a bit from what clinical practitioners are seeing. When you look at the evidence, it's not really there at the moment. So this was a great study by Kate Ackerman. She had 1,000 female athletes, but they completed an online questionnaire used to determine their energy availability status, which obviously has its own limitations. But those athletes also reported their history of iron deficiency. And what they saw was that 22% of athletes that classified as adequate energy availability had a history of iron deficiency, but a significantly greater amount, so 32% of athletes classified as low energy availability had a history of iron deficiency. So there's some epidemiological evidence to suggest this might be the case. When we're looking at sort of randomized control trials, there's not much in the literature currently. This study had only 6 male runners in it, so very small sample size. They did a crossover design of a 3-day dietary intervention where they had either 45 kilocalories or less than 20 kilocalories of energy provided to them per kilogram fat-free mass. And what you can see is that they actually saw ferritin increase as part of a low energy availability diet. And there's a few explanations for that. There's an evolutionary response that in times of starvation, we might sequester our iron and hold on to it. And that might be an acute response to what we're seeing. But really the evidence if you go through the literature, is quite mixed. So it's a bit hard to make any definitive conclusions. We put together a review a couple of years ago now, looking at potential possible pathways though for these interactions. And as Jamie really clearly pointed out, low energy availability is caused by either or the combination of a decrease in energy intake and an increase in energy expenditure. So if we link that in with iron deficiency, a low energy diet is going to have low dietary iron. So similar to the carbohydrate points that Jamie pointed about, as soon as you reduce the total food volume, you're going to reduce total iron volume as well. And that may lead to iron deficiency. They're often athletes are also training a little bit excessively to create that imbalance. So you're going to have greater exercise-induced iron losses as well. On the dietary front, this was some of the data from the Supernova 5 study that Jamie presented, just the adaptation phase. He presented the dietary data already there, but just looking at the iron intake, you can see that both the keto diet and the low energy availability diet had significantly less iron provided than a high energy diet. We also think hepcidin has a role here. We're not quite sure of the pathway by which hepcidin could be elevated. It may be an inflammatory pathway, but we're less convinced of that. We think it could be more gluconeogenic signaling and ERFI, which is a erythroferrone, which is an iron regulatory marker, usually seen to upregulate in response to altitude training, but there are some links with energy restriction that may cause hepcidin to increase. And we also know that menstrual function, if we're seeing athletes that have REDS, they probably have menstrual disturbances, which cause them to have low testosterone or low estrogen. And that in itself causes hepcidin to upregulate. So it could be multifactorial, but it's a possibility that low energy availability can increase hepcidin production. Now, in the study that Jamie talked about, the Supernova 5 study, we had a look at this as well. So similar study designed to our previous Supernova studies. We had the elite athletes undergo 6 days of the control diet to harmonize their energy availability. They did an exercise test, and then they were assigned to either the high energy diet. We had a ketogenic arm in this diet as well to compare the keto diet, and also the low energy availability diet for 6 days. And then they did a second exercise test. And here I presented, rather than the pre and post, just the percentage change in IL-6. And you can see the data I presented before is the red bar there, where the IL-6 response is significantly elevated in the ketogenic group, but both the control and the low energy availability groups didn't have any differences. So that sort of confirmed our hypothesis around it probably not being an inflammatory signal. When we looked at hepcidin, you can see the ketogenic group had a clear increase after the dietary intervention. The control group actually had a slight reduction. And even though it looks like the low energy availability group is shifting in the direction of having an elevated response, it didn't come out significant. And so that we can at this stage say it's no change. So there's no effect on hepcidin. There's a few factors that we can play around with, being the duration and the intensity of the energy restriction, which may cause subsequent changes if we were to test or conduct this experiment in a different way. But at the moment, we haven't got any evidence to support an increase in hepcidin. The final part of this figure is not to go into much detail, but just to illustrate that low energy availability, we think, has an effect on hepcidin and iron status and iron deficiency rates. But we actually think iron deficiency also contributes to some of the negative effects that are seen from low energy availability. So we think it actually might be maybe accelerating the rate of some of the symptoms are seen in reds. So for instance, we've got bone health there. We know that if you're iron deficient, you can have a decrease in bone remodeling. So if you're having the effects of low energy availability and iron deficiency, it could confound some of those effects. And there's evidence to support that pathway from multiple angles of the Rett's syndrome. So to conclude, there's theoretical evidence that links poor iron status and low energy availability. However, strong empirical research is really lacking throughout the literature. And we're currently working on a IOC project trying to really collate a lot of this evidence for that spoke of the wheel in the REDS model. And we've just found that there's nothing out there that's really conclusive at all to be able to do that. The largest effect of low energy availability may actually be through the reduction in dietary iron intake, which is chronic and going to have a long-term effect, which is normally what you see in these REDS athletes. Hepcidin may be increased as a result of LEA, but our group hasn't been able to show it in the elite athletic populations that we work with. We are going to do further studies on this and manipulate some of those means of achieving low energy availability. So hopefully, or potentially, we may see some differences still. And finally, iron deficiency may not only be caused by LEA, but may also contribute to some of the other negative health aspects of low energy availability. So we're currently thinking that if we see an athlete presenting with REDS that has an iron deficiency, it is a good idea to treat that iron deficiency in the meantime to avoid further health complications while you're also rectifying the energy deficiency. So that's all from me. My email and Twitter's there if anybody had any further questions later down the track. But any questions?
You'd kind of started off by saying that this is like what not to do, not what to do.
Yeah.
Maybe I could try and figure and ask a question about what would you do, even though that wasn't that. But in the scenario where someone is training and they might be iron deficient, is it like almost more of an indication of like the training is too much volume or too intense rather than just saying like, let's just give them more iron to bring the level back up? Like, can you think of there's a reason why you wouldn't want to have iron? Is there a reason why you're like almost putting it down as like a safety precaution?
Yeah, I definitely think there's sometimes an over treatment of iron deficiency. Not so much treating the iron deficiency, but athletes who are training think they need to take 7 iron supplements a day. when reality is they're not absorbing any of that. So if you have healthy iron stores, and when I say healthy, I mean like above 50 micrograms per liter, which is a pretty good threshold. There's no need to supplement, like enough's enough. There's no performance benefit. And I think some athletes think that you can gain more by having more. At the same time, I don't think iron deficiency is a marker of overtraining. I think adequate adaptation and adequate stimulus can still deplete your iron stores, if that makes sense. So just because you're starting to see your iron deficient doesn't mean you're overtrained or training too much. It might just mean that your dietary iron intake is not enough to meet your current acceleration in your training volume. So a good example of that is altitude training. When we go to altitude, majority of athletes will come back deficient if they haven't supplemented, even if they're healthy. It's just because the iron turnover is so large and the rate of adaptation is greater than what you would get at sea level. So you need to provide more building blocks for that adaptation to occur.
Okay.
Does that kind of make sense?
Yep. Oh yeah. And then, so whenever you're doing the serum test, is there any evidence that if you do, I mean, I don't know if you're going to convince anyone to do liver biopsy or something, but is there any evidence that you can like store the iron not in the blood, and then for that acute period where it looks like they're deficient, but it's just that you're not measuring it, but it is there in the body, but for some reason it's been like taken out of the blood, so you can't see it?
Not quite sure about that, but it's hard with iron testing because inflammation is known to just it's an acute inflammatory protein. So if you have any form of inflammation, you're going to have an elevated ferritin level and iron reading. And as soon as you start to have any factors, whether it's dietary or anything coming in training, you can start to see differences in your ferritin day to day. And we do see about an 8% flux in ferritin day to day, simply because of other factors. And so you would have to do the most invasive testing of going into the liver and the spleen and potentially other iron storage places to see if that's sort of changing day to day with different things. But I can't think of a reason why, but that's not to say I would know exactly on that front, if that makes sense.
Yep, that's what I think.
Yeah. Be cool to do though, if you can convince people to
biopsy just random tissues.
Yeah.
If I remember correctly, your supernova 5 and the low carb iron paper, that was just then. Right?
Yeah.
Yeah. Have you guys done any studies looking at sex differences there?
Yeah, it's something we've moved right into now. We've got studies on the go. We have a rugby league. I don't know if that's Canadian or not, but we have a big bunch of female athletes coming down to do a training camp with us in August. And Supernova 7, as Jamie mentioned, is going to be completely in female athletes. We're struggling, or have struggled in the past, because we obviously know the menstrual cycle has a huge effect on iron. And if you have a 4 to 5 week training camp, how do you control every girl on that training camp cycle? So female studies are so hard and they're so important, but they don't suit the training camp model really well at the moment. And we're working through how we can sort of adapt things or monitor things better so we can account for it. I do think there's sex-specific differences. We see generally during when an athlete has their period, you'll see a huge decline in ferritin straight away, and hepsin will follow, and then it will restore and probably overcompensate a bit at the end of the cycle. So within everything you're already looking at, you're seeing these like micro changes in ferritin and hepcidin, which make it complicated to study. Outside of that, we think the physiology and the iron regulatory pathways are pretty similar between the sexes. Yeah.
With all the pathways and mechanisms you got into, it almost seems like it's impossible to keep your iron. But like, I think the highest figure you gave was 35% in women being iron deficient. So what are, if people who are having success keeping their iron, what's making them different?
Yeah, it's a good question. I think it's really individual, and we know there's genetic aspects as well to iron that we don't really touch in our studies being athletes. We look at not what athlete has what gene, but rather athletes as a whole. The other thing which makes those numbers really hard to interpret is that if you're a high level athlete and you're working with a sports scientist or somebody like myself, you're not going to be in an iron deficient state. You're going to be corrected before you get into that, or monitored better, or have dietitian support behind you. If you look at some of the sub-elite or the highly trained athletes, you often see the highest incidence of iron deficiency in those guys. because they have no idea they're iron deficient, and it can go on for a long time before being picked up as iron deficient. The prevalence rates are really hard to interpret, I think I would say with caution. And a lot of athletes that when I was working within the institute system that would come through, you get on top of it early when they're young. If they're at higher risk of iron deficiency, you can kind of tell when somebody's within certain zones, and therefore it's a bit easier to manage. But if that makes sense, like the highest deficiency rates are usually in those that aren't being screened, and in the elite population studies that have been done, which those figures are drawn through, are athletes that are looked after a bit better. So iron deficiency is known to be the most prevalent deficiency worldwide. I just think the athletes we look at are probably a little bit better cared for than other people that are exercising in the general community.
Thank you.
Cool.
Okay, thank you so much for the great talk. We have our last but not least speaker today. So we're going to hear from Dr. Nolan Hoffman, and he's talking about human skeletal muscle signaling networks underlying high intensity interval training. So Dr. Hoffman completed a Ph.D. at Indiana University School of Medicine in 2012. He had a 2-year stint as a postdoctoral research associate at the University of Sydney, and is now being a research fellow at the Australian Catholic University since 2016.
Great. Thanks very much for the introduction. And first of all, thank you very much to the University of Guelph team for hosting us today. It's been great being back in North America and also seeing Jamie's stomping grounds that we hear so much about. So very nice to be up here in Guelph. So today I'll shift focus a little bit and tell you about some of the more molecular work that we do down in Melbourne in the Mary Mackillop Institute. So as was introduced, so I'm involved with several aspects, really from the cellular and molecular side up to the human side. So my background, so I did my PhD in cellular and integrated physiology. So at that time, I was mostly working with cells, started doing some animal studies, and in my first postdoc worked a lot with animal and started collaborating with some labs that were using exercise models. So I'll have a disclaimer at the beginning of my talk. I'm not an exercise physiologist. My background's in molecular metabolism, but what we really do is work together with exercise physiologists such as Jamie and Alana to use exercise as a tool to really map a lot of these molecular networks that are involved in the basic biology of exercise. So today I'll talk about one theme of my research that we work on in our group. So one major theme is molecular networks underlying exercise, which will be the focus of my talk today. And just for the sake, because I know a lot of people do other work around here that's very relevant to what we do. So the other theme that we work on is energy sensing and availability. So many of you might be familiar with AMP kinase. So we have several transgenic mouse models that we work on with AMPK, but today I'll really focus on the human side and really show you how we use this integrative approach to really work from the cellular models all the way up to using human models to understand basic biology. So just to start my talk, all of you will be familiar with HIIT, or high intensity interval training. And just from this nice figure from McGinnis and Marty Gabala, some local leaders in HIIT biology here in Toronto. Many of you will be familiar with all the beneficial effects of exercise and HIIT specifically. And we know, especially in skeletal muscle, we have cellular energy stress that's induced with exercise. And this leads to many beneficial physiological adaptations, such as increasing mitochondrial content. And when we think about different forms of exercise, we know that HIIT can be equivalent or superior in some respects to inducing these molecular adaptations. However, it's important to note that to date, there's not really much known on the molecular side of what are the signals that actually underlie these unique benefits. So the focus of my talk today will be on skeletal muscle, and I'll introduce you to a project called the HIIT-OME study, where we've utilized omics approaches and human skeletal muscle biology to look at these molecular networks that may underlie some of these benefits of HIIT. So as the focus of my talk today will be on omics, for those of you who aren't familiar, we have several different biological layers in every cell throughout our body that really program and allow us to engage, for example, biological benefits to our metabolism, for example, with exercise. So there's many different layers. So if you look on those research tracks on the top right, so from our building blocks in the genome, all the way up to how our genes are exposed to our environment with the epigenome and how these genes are transcribed. and translated eventually to proteins. There's many different layers where you can control the biology in response to acute exercise. But what I'm really focused on is signaling networks. So if you haven't been exposed too much to the phosphoproteomics side, today I'll tell you about how we're using a specific form of proteomics called phosphoproteomics, where we can look specifically at how proteins are chemically modified in response to, for example, exercise to elicit some of these benefits. And as you'll see on this slide, we know there's many different variables that can influence how these molecular networks are engaged with exercise. So the focus of my talk today will be on mode and intensity of exercise, but just keep in mind there's a lot more complexity to it. So we have many different cell types that are engaged with exercise, and many of them can crosstalk either to different cells or different organs. And we also have several different subject criteria that Jamie and Alana have touched on, such as sex, age, and underlying all this is genetic variability, and also with the nutrition status and training status. So the first two talks have been more on the elite athlete side. So I'll shift the focus today more to the general population that we use to study a lot of this basic biology going on with exercise. So as I hinted earlier in my talk, so we know that HIIT's beneficial, but what are the molecular mechanisms underlying this? So there's been many different proposed cellular mechanisms involved. So for example, increased ATP turnover, increased glycogenolysis, so potential effects. So when we think about HIIT, we have high intensity exercise separated by bouts of rest or active recovery periods. And we think that this buildup of calcium and other metabolites in the cell may influence the molecular networks in signaling events that are engaged. So there at the central on panel E is signaling cascade activation. And that's really what we're getting at in this project, to see what are the signals engaged with muscle, with HIIT versus moderate intensity continuous exercise, or MICT, that elicit some of these benefits. So hopefully today we can fill in some of these gaps in some of the knowledge underlying this biology. So first, let's talk about phosphorylation. So why is protein phosphorylation important in exercise biology? So we've talked a lot about nutrition, iron status. So if we think about the basic biology within a muscle cell, so we know with exercise, we have these switch mechanisms. So you can think of protein phosphorylation as a switch mechanism that is able to be switched on or off with exercise. And phosphorylation really lies at the heart of mostly all biological processes throughout the body, especially when we think about exercise stimulated responses in skeletal muscle. So when we think about the switch mechanism, okay, how do we turn that switch on or off? So that's using what's called a protein kinase. So we have kinases, for example, such as AMPK, that might get engaged by, let's say, cellular energy stress, get turned on, and those phosphorylate what are called downstream substrates. So one protein can recognize different proteins throughout the cell and phosphorylate them and regulate downstream biological processes. So you can think of it as a signal output functioning on the switch mechanism leading to signaling outputs. So why is this important, and why would we want to control these switches? So we know that phosphorylation can regulate many aspects of cell biology. So for example, it can change the way a protein becomes activated. So let's say a protein gets phosphorylated on this certain amino acid that can make it active. It can also change the localization of a protein in the cell, or maybe its conformation or binding to other proteins. So you can think about it as this intricate network of switches in every cell that we can light up, for example, with exercise. So that brings me to the HIIT trial, where we've tried to map this with high intensity exercise versus MICT. So the primary aim of this study was to determine the effects of a single session of work-matched high intensity interval training versus MICT on the human skeletal muscle signaling network. And I'll walk you through this trial. But what we do is we have a series of baseline measurements before the participants start. And then in this study, we recruited 10 participants who are relatively physically inactive, but otherwise healthy and disease-free. So you can think of typical university-age students. So we recruit them to do 2 forms of exercise separated by about 10 days between the 2 forms. So if I walk you through this figure, so when they come in, they do preliminary testing, which I'll go through in a minute. And then we perform skeletal muscle biopsy collections with the help of our Dr. Andrew Garnham. So we collect a muscle biopsy at rest, so pre-exercise, and then we collect a muscle biopsy during exercise, which essentially, if you think of HIIT, during one of the intervals, we essentially have a bed behind the bike, and the participant falls back on the bed with some help from us, of course. And then we take a biopsy during exercise, and then they essentially get back on the bike. We help them start the pedals, they get going. And then so they have the 0 minute pre-exercise, 5 minute during exercise. And then at the end of this 10-minute bout of exercise, we take a post-exercise biopsy. So the goal with that is we know phosphorylation happens really quickly. You might wonder, why are we only doing 10 minutes of exercise? That's how rapid this occurs. So we know within 5-10 minutes, a lot of these proteins are changing. And just to look at more of the downstream responses, we also collect muscle biopsies at 1 and 3 hours post-exercise. So let's go through some of this preliminary testing first. So if we think about the inclusion criteria, so as I mentioned, we're not testing elite athletes. We have to start, if we don't know what's happening with the basic biology here, you really have to start with a healthy, untrained participant, because it's well appreciated that the more trained you are, the less signaling responses you actually see in muscle with exercise. So if we were going to test some of the elite athletes, for example, that Jamie and Alana test, we probably wouldn't see that much going on because they have such fine-tuned systems there. Or if you think about AMPK, it's already so active that you can't really budget that much more with exercise. So these participants that we recruited were males aged 18 to 30 years old with a healthy BMI. And when we think about their physical activity levels, so they're not sedentary per se, but they're doing lower than the recommended level of physical activity. So we consider that less than about 150 minutes per week of moderate intensity exercise. Otherwise, we do some general health screening to make sure they don't have any major cardiopulmonary abnormalities, no cardiovascular disease or diabetes, no bleeding disorders, because we're obviously taking muscle biopsies and blood samples from these participants, and then any major or chronic illness that impairs their digestion. And finally, we just do a background history about previous prescription medications, just to make sure they aren't taking anything that may influence signaling networks. So if I first go through the baseline measurements, Jamie and Alana have already touched on a lot of this, but just briefly, so we do a DEXA when they come in the fasted state. On the same day, we do a resting metabolic rate test. And really, this is just to get a measurement of their resting metabolic rate and helps us prescribe these standardized meals that they'll take before every exercise bout. And then we do a VO2 max test with the help of my good friend Jamie here. And then following the VO2 max testing, then we're able to prescribe the exercise that they're going to be doing. So I'll quickly just walk you through what these participants look like. So if we think about their baseline characteristics, so these participants were on average about 25 years old, had a BMI healthy range about 23.5, and they were on average about 75 kilos. Again, we had these 10 participants, and as you can see, we take several different blood measurements before just to get an idea of their health status. So once these participants are recruited, with the help of Jamie, we prescribe the exercise modalities. So something really important to note in this study, when you look at the hit and mix studies that are out there, it's a bit of a mixed bag. Some use similar workload, similar duration, but others you're really comparing apples to oranges. So in this study design, what we wanted to do was control everything possible so that by the end of the 10 minutes of exercise, they're doing exactly the same workload and over the exact total duration, which is 10 minutes. So using this experimental design, so these participants had a relative VO2 max of about 38, which is pretty characteristic of what we see in our relative untrained university-age males on campus and in the general Melbourne community. And then we prescribe their HIIT power outputs. So as you can see, as I mentioned, they do this interval form of exercise where on their on intervals, they're doing about 175 watts. And that's about 85% of their maximal workload. And then on the off intervals, they essentially just have an active recovery period where they're cycling at about 50 watts. So that we can balance the workload, we prescribe a workload for the moderate intensity that was equivalent of about 55% of the work max. And this is corresponding to about 113 watts. So if you just look at some blood parameters first, so we take blood samples at 0, 5, 10 minutes. As you can see, there's no major changes in glucose, but if we look at the lactate, we see a nice increase over time. So lactate increasing at 5 and 10 minutes. And as you see with the HIIT response, there was a main effect of exercise mode. So more lactate is being produced in response to the HIIT. And remember, this is the crossover design. So the same participants doing the same, doing each exercise bout separated by about 10 days. So a lot of this work is going to be using omics. So this is really branching on a lot of the work that I did in my first postdoc with David James in Sydney. So I'll first introduce you to phosphoproteomics, if you're not familiar. So as I mentioned, we have all these proteins in our cells. Many of them can get phosphorylated. And phosphoproteomics essentially uses an instrument called a mass spec or mass spectrometer. And this allows us to map, identify, and quantify all these proteins and phosphorylation events. So I like to think about it as, imagine you have a puzzle with all these puzzle pieces. You throw them in the mass spec. The mass spec can basically identify each of those puzzle pieces and then put them back together to tell you what protein that is and also where was the protein phosphorylated on which amino acid. So in this first study we did from my previous work in Sydney, so this was the first study to ever look at human exercise phosphorylation networks in skeletal muscle. And in this study, just one single bout of exercise, about 10 minutes on the bike, we see about increased about 1,000 exercise regulated phosphorylation sites. So it's not as simple as just looking at a couple proteins. We're talking thousands of phosphorylation events here. So just to give you an overview of the phosphoproteomics workflow we used in this study. So for the sake of time, I won't go through everything, but I'm happy to answer questions after if you want more details. But essentially, we take the skeletal muscle biopsies. We essentially snap freeze those as quickly as possible because we want to preserve all those phosphorylation events that happen in the muscle. And then from that, we basically take the frozen muscle and we extract the proteins. And then from those proteins, we essentially chop them up into peptides using trypsin. So after that, we do a few chemical procedures for the sample preparation just to get all those tryptic peptides ready for labeling and enrichment. So essentially with these labels, we use essentially what's called a tandem mass tag isobaric label. So you can think about it as a little chemical tag on each peptide, and it allows us to put in, like we use a 10-plex tandem mass tag kit, which you can essentially have 10 samples, label them all differently, put them together, and the mass spec can recognize all those chemical tags and tell you which sample it came from. Then we can do what's called phosphopeptide enrichment. So as I mentioned, we're not just interested in what the proteins are, but how they're phosphorylated or chemically modified. So we use what are called titanium dioxide beads, and essentially the phosphopeptides bind to that. So we can collect those in a test tube and then basically fractionate them out, and they're ready to go on this instrument at the bottom called a mass spectrometer. And essentially inside this mass spectrometer, you're identifying basically every minute thousands of peptides and phosphorylation events. So with omics and exercise biology, there's many hurdles we have to overcome. When we think about, the training camps, for example, the huge hurdles, subject recruitment, getting all these participants through and getting all their diets prescribed. So in this case, it's a little bit different. So the subject recruitment's a huge part of the study, but really the challenging hurdles come later on where biologists have to work with mathematicians and we have to put all this complex data together. So I'll introduce you to some bioinformatic workflows that we use to basically analyze these data throughout my talk. And essentially a big part of this, so after we basically prepare the samples, get them run on the mass spec, then we do bioinformatics, which you can think of as a statistical workflow that's looking at, instead of a few rows on a spreadsheet, like tens of thousands of rows. And then we can visualize these data because it's not going to help us much if we just try to publish, for example, a 10,000 row spreadsheet. It doesn't tell us much. So we have to come up with unique ways to visualize these data and make it digestible for us, but also the general biological community. And really the last step of that, we're ideally picking out a few of those and validating them. So let me show you some of the overall data, and we'll just go through the broad brushstrokes before we get into what happened with exercise. So like I said, we have 10 participants. We have 3 time points, so 0, 5, and 10 minutes, which I'll refer to as pre, mid, and post exercise. We have our 2 exercise modes. So essentially on the mass spec, we had 60 samples total. So in the raw data set, so you get this horrible looking Excel file back that you work with the bioinformaticist to make sense of everything. And like I said, put those puzzle pieces back together. So there's specialized software that you can use that's connected with the mass spec that basically puts these puzzle pieces back together, tells you what it is. And then we work on doing the stats with the bioinformatics team. So in the raw data set, we had roughly 15,000 phosphorylation sites that we identified just in skeletal muscle. And I'll comment, skeletal muscle is a pretty tricky tissue to work with compared to other tissues We know that there's a lot of structural proteins, like one, for example, titin. I spent about the first year of my postdoc trying to optimize skeletal muscle proteomics. And I think the first several runs, we only identified titin, and it's like 10,000 kilodaltons. It's a huge protein. So you have to be careful when you're preparing these samples in such a way and fractionate them out, so you can basically see through the clouds and not just the cloud itself. So once we saw through the cloud, essentially, we had about 15,000 sites that corresponded to about 12,000 that had a localization probability of 75% or above. So we basically want to make sure that the mass spec is confident at what it identified. So we set a confidence interval for that one. And then we want to look at sites that occur in multiple participants. So in this particular study, we made sure that any site we're talking about is identified in at least 3 of the 10 participants. So to analyze all this data, like I said, we work with bioinformatics team. So in this particular study, it involved Ping Yang, who's my long-term collaborator based in Sydney, and Veronica Sumi, who was at ACU. And we're using this workflow called Phosphor, which we published last year in Cell Reports. And it's essentially a proteomics workflow where instead of having to go and search for all these scripts separately, we've essentially packaged everything together. So if you have a phosphoproteomics data set, you can start pumping it through this pathway, and it'll help visualize your data and analyze it. So I'll show you some of the visualization of this. So some of these plots you may not be familiar with, so I'll just walk you through them. And I'll try to give you take-home messages along the way to maybe make it a little more digestible. So first, typically when you get large data sets like this, you do what's called hierarchical clustering, which basically tells you how related are each of the samples to each other. So which ones are more similar to each other. And what we were really excited to see is our level of biological control. So if you look on the left side, so you're just seeing the purple and the green samples, which are the pre from each exercise trial. So that basically tells us that the baseline profile is very similar to each other, and that with our dietary control, we do the days leading up and basically tell them not to exercise the days leading up and everything, that we were able to have a relatively consistent control sample. And then following this, we looked at how the samples cluster, so mid and post each exercise bout. And excitingly, the profiles of the MICT versus the HIT were classified very differently and further apart. So if you notice, for example, the red and the yellow are separate from the 2 blues, which correspond to the HIT. So then if we visualize this using principal component analysis, so you can think of each dot a sample, and essentially each dot will tell you how related or how different it is to the other samples. So as you can see on the left here, so the pre-samples clustered very closely together, as we would expect. And then in response to exercise, we see this shift in the overall profile of the data set. So as you can see, the blue dots are very far away along principal component 1, suggesting that there's a response in the phosphoproteome to exercise. And then excitingly, the MICT were very, very different clusters than the HIT. So this suggests that each form of exercise, even the same time duration, there's something about those intervals that's leading to a different signaling profile. So just to wrap up some of the data and look at some of the final numbers. So first we wanted to see, okay, which sites were different with HIT and which sites were different from MICT. So this isn't comparing the 2 forms of exercise yet. We just look at them in isolation. And as you can see, just after hopping on the bike for 5 minutes, there's already nearly 1,600 sites that are already significantly regulated. And when we say significance for these sites, that's basically just referring to a fold change greater than 1.5. So increase of 50% or more phosphorylation, either up or down, and then also an adjusted p-value of less than 0.05. So if we look at the HIT first, so like I said, about 1,600 or so up at 5 minutes. So this stuff happens really quick. And then at 10 minutes, we see about 2,000 sites with HIT. And similarly for MICT, we see about over 1,000 sites that are increased at 5 or 10 minutes. And if we next visualize these, so this is what's called a volcano plot. And along the x-axis, we're looking at the log2 fold change. And along the y-axis, the minus log 10 p-value. So essentially, if you just focus on the middle of the top left, for example, if you're looking at each dot represents a site. So on the right, the red dots are sites that are increased, for example, with exercise, and then the blue ones are decreased with exercise. And then above the line here. So anything above that's not black in the background is a significant site. So that just visualizes those 1,000 sites. So you see already at 5 and 10 minutes, a lot going on with HIT and similar with MICT. So next, we wanted to ask the question, which signaling networks are potentially different between these two forms of exercise? And this is really our primary aim of the study. And just I'll highlight really quickly on these volcano plots. So I just didn't want to show you a bunch of dots, but what we can look at is essentially, I think this is just showing the top 10 up and the top 10 down. So, and these are essentially the gene name, and then you'll see a semicolon followed by the amino acid residue. So we can start to look at some of the top regulated sites, but there's some really cool ones in there, such as some mitochondrial fission processes going on and a lot of new biology that we wouldn't really expect. So it'll be really interesting to follow this up in the years ahead. So next, let's look at the HIT versus MICT. So we weren't sure going into this, is exercise just going to stimulate phosphorylation of all these proteins, or are we going to see anything different? But it was really exciting to see several phosphorylation sites popping up that are unique to HIT. So if we look on the left at 5 minutes, we start to see a few, so about 92 total sites that are significantly different between HIT versus MICT. And then as you continue exercise up to 10 minutes, we start to see hundreds of sites that are different between HIT and MICT. So just that interval alone is leading to these different signaling events. So the take-home message for this is essentially HIT stimulates a new unique network of muscle signaling responses versus MICT. So this will really be the focus of our follow-up work in the years ahead to start hopefully getting a lot of grant funding to study some of these new ones. But I guess it's important to note that we would need basically an army of PhD students to study these 100 sites. I mean, in my first postdoc, to study the biological function of one and overexpress it, knock it down, mutate it, it can take up to years to just find the function of a new site alone. So it does take a long time to follow up a lot of this work, but validation is really important because we found this new biology, so we want to find the function of it. So next, if we look at those hundreds of sites that are different between HIT and MICT, let's look at some of the biological pathways that were different between some of them and also some of them that are similar. So a lot of this is going to be too small for either the audience or the people online, but just to summarize, we see a lot of the pathways that we would expect to be up with exercise. So for example, on the very far left, you see mitochondrial transcription, which we see above the dotted line there for the Z score, above about 1.5. That suggests that mitochondrial transcription is up, for example, with HIT, and we don't see that with MICT. So there's quite a few pathways that we expect to be up or down with exercise that we detect in our data set. So I don't like to take too much information from these pathway analysis. It's just more about storytelling and maybe guiding you on a particular pathway that might be interesting. But what interests me most about this data set is kinase activity. So as I mentioned, kinases have the ability to phosphorylate all these substrates around the cell. So what we can do is use kinases and their known substrates to start to map within our data set what's happening. So I've tried to highlight a few that may be familiar for you that we can talk about in more detail. So let's first talk about pathways that we know are increased with exercise. So AMPK is a perfect example. We know it increases in phosphorylation with most forms of exercise, and that's exactly what we see here. So above that dotted line, we see AMPK activity increased when we look at its known substrates across the data set. And on the far right, so we know with mTOR is a kinase that's traditionally activated by resistance exercise, but not endurance or aerobic exercise. So in this data set, that's exactly what we see. We see AMPK up, mTOR down. That's what we would expect. But what's really interesting is we start to dig into some of these kinases that are different. So for example, one of the follow-up work that I'll be focusing on is PKC isoforms. So we see some distinct PKC isoforms that are responsive to either HIT or MICT. And that will be the focus of some of our work. And there's also several other kinases that haven't really been implicated in exercise that we see up that are potentially hinting at new biology in response to exercise. And as I mentioned, validation is an important step of this. So what we can do is go in the lab and take those exact same muscle samples and start to do some Western blotting to confirm what we saw in the mass spec. And that's exactly what we see here. So for example, with AMPK, we see it increased in phosphorylation on the activation residue and also on its downstream substrate in response to both forms of exercise. And just in the last part of my talk, I'll just focus on visualization and show you some of the other things we can do with this type of data. So I mentioned we know we have kinases and they have this known set of substrates. So what we can do is start to map out different substrates. So let's say, for example, with one of the PKC isoforms, we can look at its known substrates and see what's going on with each form of exercise. And that will really help us go into the lab and either use antibodies that we can purchase commercially or create new antibodies where we can essentially detect these new phosphorylation sites and uncover that new biology. Also, what we can do is predict new kinases and substrates. So a large part of my work through the years is, for example, with AMPK substrates. So finding new substrates of AMPK and maybe pathways that we didn't know AMPK regulated. So we can use these predictions to look at, okay, in this heat map, for example, how likely a row, which is a site, is phosphorylated by a given kinase or a column. And as you can see with HIT versus MICT, you start to see a few different patterns emerge. But this will be the focus of some follow-up work where we look at how these kinases may be differentially regulated with different forms of exercise. And finally, we can start to look at the kinase and connect it to the pathway. So a couple of good examples here, which I've tried to highlight in red. So along the x-axis are kinases, and along the y-axis are biological pathways, for example, with using Reactome database. So a good example is GSK3 beta. So if you look at MICT first, it's all in the purple, suggesting it's not really regulating any of these pathways. But all of a sudden with HIT exercise, the GSK3 beta is activating MAPK pathways. So there's several different unique aspects of this. And if we look on the far right, so we see other PKC isoforms that seem to be hitting different pathways. So there's something interesting about those intervals that's leading to different biological pathways. And finally, we can start to package all this together into what we call signalome networks. So imagine taking a kinase, its known substrates, and packaging all together into almost a hairball or a web diagram like this. But essentially what we can look at within this hairball is essentially HIT protein modules versus MICT protein modules. So for example, let's say this phosphorylation site here on the left in module 2. So this is about 200 proteins in the data set. With HIT exercise, you see a huge decrease in phosphorylation, whereas it's less robust with the MICT. Or for example, with HIT, you see something that comes up at about 5 minutes and gets turned off, whereas with MICT, it stays up. So there's several interesting potential pathways in here. And like I said, hopefully will be the gift that keeps giving with student projects through the years. And basically to summarize some of this, the HIT and MICT exercise signaling networks have shown kinases that can each phosphorylate unique substrates in also different directions. So increasing substrates, decreasing substrates. And finally, just putting all this data together. So hopefully I've shown you today some of the work that we can do using omics and applying it to exercise biology to find new pathways and hopefully new mechanisms that promote health and performance. And within these unique pathways, we see kinases and substrates that seem to be very specific to HIT and hopefully will help us uncover some of the unique muscle adaptations that occur in response to HIT. And just like to thank my colleagues, especially the exercise physiologists who help out and make this work possible, and my bioinformatics team and mass spectrometry team at the University of Melbourne and University of Sydney, and our funding through the Australian government and ACU. Thank you very much.
Thanks for your talk. It's very impressive. The amount of data you can generate from those results. You showed some decrease in phosphorylation size of after exercise, which suggests you spoke about a lot about the kinase side of things, but decreasing phosphorylation levels suggest increasing phosphatase activity. So, like, I was going to ask about if you use the enrichment analysis to see if the increase or decrease of phosphorylation levels is based on which residue you're looking for, like serine or threonine, or maybe if you look at the enrichment analysis more based on the signaling you're talking about.
Right. Yeah, it's a great question. So I guess to answer the second half of your question first. So in our data set, we identify basically amino acids serine, threonine, and tyrosine that are phosphorylated. So all three that can potentially become phosphorylated. And then in our data set, so our key question is which sites get upregulated or downregulated with acute exercise. So like exactly right, it's a great question. So it's not just kinases, on the balance is phosphatases. So I talked a lot about kinases, and in response to exercise, we know we can activate a lot of those. But as you saw in the data set, a lot of sites decrease, which would suggest that in the basal state, they may have a high level of phosphorylation, which may be active in the resting state. But then in response to exercise, we turn off some of those pathways. And we really have to do that, for example, to make sure. So in response to exercise, we know we need to increase glucose transport, for example, and phosphorylate a lot of insulin signaling proteins. But also after a certain amount of exercise, we don't want to keep activating those same proteins and, for example, cause low blood sugar over time. So there's a lot of feedback mechanisms that occur. So a lot of what we're seeing in response to exercise is kinase activation, but also phosphatase activation or deactivation. The databases out there aren't quite as good for phosphatases, so we use a lot of what's known about kinases. But I'd say there's a lot more we can do in the years ahead once there's more and more biological evidence to say, okay, that site is regulated by that phosphatase. So in response to exercise, that phosphatase must be deactivated or something. So I think there's a lot more work we could do on the flip side with phosphatases. It's a great question.
Yeah, and you did some biopsies in two moments of exercise and then after exercise. Can you combine your proteomics, I'm thinking about the transcription factors, with some RNA-seq to actually target which genes are being up or down regulated based on the activation of the
Yeah, definitely. And that's a huge growing field, the multi-omics space, or integrating multiple omics, because we know, for example, phosphorylation I mentioned is lying at the heart of a lot of biological processes, but phosphorylation also is really important for regulating transcriptional activity. So there's a lot known, and there's a few good databases out there where you can kind of map a kinase or a phosphorylation site to a transcription factor. So for example, in the first study we did of exercise in human muscle, we actually identified quite a few transcription factors that get phosphorylated themselves. Unfortunately, a lot of them aren't very abundant in muscle. So a lot of times when you look at the whole data set, you might not see that many transcription factors there. So exactly like you suggested, what you can do is do RNA sequencing or another omics platform and hopefully integrate those two together to say, okay, we see this gene going up and that site going down that regulates that gene. So it'll connect the dots essentially. So I think hopefully with the funding, we may be able to do more omics layers. But given that signaling is really the focus of what I do, and I definitely work a bit more on the protein than the gene side to look at the functional responses downstream. But I think there's a lot more we could do with the transcriptomics. And we're definitely open to collaborations such as that, especially with the 1 and 3 hour post samples we haven't really done much with yet. So I think there's a lot of value for potentially looking at the transcriptomics profiles that, because they don't occur quite as rapidly as protein phosphorylation, but in the hours post-exercise, you can definitely see those lighting up. So, and we have a project going on with David Bishop's group right now, looking at essentially the networks that are involved with these transcription time courses over time. So hopefully more to come on that in the years ahead.
I'm just wondering, are you able to apply that same workflow to other post-translational modifications other than phosphorylation?
Yeah, it should be applicable. So in this case, we focus on phosphorylation, but it should be applicable to essentially acetylation. There's also ubiquitination and a few other post-translational modifications that occur. So I don't know the exact scripts that you would use to do that, but essentially you would, the column that's telling you the phosphorylation site, you'd basically just replace that with the acetylated one. And because we don't enrich for acetylation in our data set, we don't see a lot of that. I think you really have to take quite a bit of muscle enrich for acetylation to see a lot going on, especially if you use an omics approach. Because the few studies that are out there, they've been able to detect a few acetylation markers of exercise, but I think you need quite a bit of sample. So there's definitely capability to use that workflow later on to do stuff like that. Yeah.
I got a question. I apologize if it was in your talk.
Yeah, it's right.
It's hard to hear. You have a 5-minute biopsy and 10-minute biopsy with your head. Is that immediately after exercise or in the recovery period for the 5 minutes?
Yeah, so 5 minutes is during exercise.
Yeah.
So essentially, yeah, we have a bed at the back of the bike. So it's easy on the hit because we do it during
Yep,
one of the
what
off
about 10
intervals.
minutes?
Yeah, and so 10 minutes immediately post.
Okay.
So
So
yep.
you kind of set it up where you've got moderate
Mhm.
versus hit, and hit is at the high end. So you have no idea what's happening in the recovery.
Exactly.
period.
Yep.
And I mean, your interpretations have all been about this is specific to hit. And I'm
Mhm.
always worried about whether or not it's just specific to phosphocreatine use and activation of purine nucleotide cycles and all the other signaling cascades that correspond to that.
Yep.
So what do you think would happen if you just went 2 hours to fatigue at this 117 watt moderate intensity? Would you actually get differences in the proteomic approaches?
Well, it's a good question. We typically, I think most of the studies we've done have kind of hovered around the 10-minute, 20-minute. I think the longest we've gone is 30 minutes so far. But because this field's so new, nobody's ever gone past. So it's basically an unopened Pandora's box there. So I agree, there's probably a lot of biological processes that are contributing. It's not just the signaling, there's all these other pathways such as phosphocreatine feeding into this. So I think if I had to guess, if you just put one of these participants on for 2 hours constant, you would probably see like typically AMPK activation in our hands with omics data probably occurs between 5 to let's say 30 minutes. You might still see it turned on at 60 minutes. But I think in these participants, given they're so untrained, you may still see a little residual activity at 2 hours, but we kind of look at that peak activation. Yeah, because each kinase kind of has its own time course. So AMPK, we consider like an early kinase, whereas mTOR and others sometimes take a bit longer to activate. So, but yeah, that's a great question. I think there's a lot more we can do to look at the time course. So one thing we're doing currently, which I feel a little bad for the participants, but we're doing a study with essentially 12 biopsy time course for that transcriptomic study I mentioned. And essentially they get a biopsy 0, 10, 20 minutes, 1 hour, 3 hour, 5 hour, 7, 9, 12. So we have a nice time profile there. So actually we could answer that question. We'd have to get the funding to look at that many samples though, because unfortunately, like with this study, so about 60 samples, it extrapolates pretty quickly on the budget. So,
but how long are they exercising in that study?
Oh, good question. So in that study, I think they're exercising 30 minutes total. So we would get that.
This isn't just you. Everybody with HIT does it, right? HIT goes to fatigue, and moderate is always volume max. And by default now, it looks like HIT is superior for VO2 max training, for mitochondrial biogenesis, for all the signaling cascades. But if you go back 15 years ago, they weren't volume matched and everything was the same.
Right.
So all your signaling cascades, everything P38, you name it, if you go back to some of the original work, they weren't volume matched. It was, I'm going to do 2 hours of endurance or an hour endurance because that's actually endurance training. We would never tell people to do 10 minutes of low moderate activity and call it endurance training. We'd say let's go in for a walk.
Yeah. So with this study, I'll be honest, we use the jackhammer approach, as I call it, to basically use trained subjects, train them or basically exercise them for a small amount, see what gets turned on. But I think there's a lot of value that can be done with, and I think there's more and more data. Like there's some recent stuff from Bishop's group actually that shows kind of some of the HIT versus, I think it's SIT versus MICT, but they look at some of the volume versus the training load, and they start to pick about some of these differences. But on the acute side, I'm not quite as sure. It's pretty early stages. Great. Well, thanks very much, everyone.
Thank you.
We're also looking for a postdoc. So if any of you are PhD students, we're current, not me, but John is currently recruiting for a postdoc. So if anybody's interested, please do.